Now?
Yes, ma'am. Started.
Yes.
Okay, good. So, the reason why I'm asking you this is because the second unit is all about structure and function of microbial cells, and we'll be talking about prokaryotes and eukaryotes. So this is the reason I wanted to know कि कितना उसमें बता दिया गया so that there's not much of an overlap. Um, anyway, so bacteria नहीं बताया है तो we'll today start with the prokaryotic cell. And who is the coordinator of the course? Who's taking the lectures right now for biological sciences so that I can coordinate?
हम अर्चना चुग
Archana, ma'am.
Archana ma'am. Okay. ठीक है, fine. स्कूल टाइम में तुम लोगों ने यूकैरियोटिक सेल पढ़ा है, राइट? सेल स्ट्रक्चर पढ़ा है ना?
Yes, ma'am.
लेट्स से कौन बोल रहा है?
मैम 10th तक ही पढ़ा था.
10 तक पढ़ा है
अभी
ना? तो सेल तो पता है ना क्या होता है? सेल के स्ट्रक्चर जैसे माइटोकॉन्ड्रिया तुम लोग मुझे बता रहे थे कि इट इज द पावर हाउस ऑफ सेल, ये तो पढ़ा है ना?
यस मैम 8th में.
8th में पढ़ा है, ठीक है. तो मतलब इसका क्या मतलब है कि भूल गए हो अभिनव?
नहीं मैम, याद है तभी तो बोला था.
याद है ना? ओके. ठीक है. तो we will first recapitulate what you might already know. In this unit, which I plan to cover in the next 4 or 5 lectures, I will be talking about both prokaryotic cells and eukaryotic cells, and then I will take up some representatives to show you the structure, life cycle of let's say fungi and protists and stuff. Bacteria will be the main focus for the entire course. And in this context, we will also talk about archaea, some of the things you already know from today's class, it seems. अब तुम लोग पहले मुझे बताओ, you all are aware of prokaryotes and eukaryotes, right? You know what
Yes,
are
ma'am.
prokaryotes and eukaryotes, huh? So, like I said, it's just going to be just a recapitulation. What you mean by the term here is karyon refers to seed or the nucleus. And when you say prokaryote, pro here refers to primitive and eu refers to true. So the name would suggest that eukaryotes have a true nucleus. और true nucleus का मतलब क्या होता है? True nucleus मतलब where all the genetic material is stored. Okay. So this is kind of well formed in the case of eukaryotes, while prokaryotes have a primitive form of it. By primitive, I mean that the genetic material is not contained in the form of a well-defined nucleus, which you find in a eukaryote. Now there are two kinds of organisms that fall under prokaryotes, bacteria and archaea. और तुम लोगों ने archaea में शुरू किया है from in your biology lectures. So I'll take up bacteria first, and then I would like to know कि archaea में कहाँ तक cover किया, so that I can, for me, archaea would cover the topic. Archaea would cover the major differences between bacteria and archaea. I'm trying to explain to you how archaea are so successful in extremes of conditions. So why are they called extremophiles? That will be my focus. And if it has not been covered in biology, I would like to cover it. Otherwise, we can skip this. Then in eukaryotes, you have all the other groups like algae, fungi, protists. That is majorly the higher organisms, plants and animals. They are all eukaryotic. Okay. In itself has certain common features that they share. So both prokaryotes and eukaryotes share certain common features. Bolo? First of such common feature, the most important one is of course that there has to be a genetic material. It is considered. Please mute करो अपने आप को. क्या हुआ? हम करें? यतीक सिंह. The DNA or the genetic material is the blueprint of the cell, and it stores all information about the cell's potential. You all know this. This is DNA is a deoxyribonucleic acid. Besides the DNA, every cell has to have an outer covering, which is called the cell membrane. अब हम लोग हर एक के बारे में बात करेंगे डिटेल में. अभी हम आपको सिर्फ बता रहे हैं कि कॉमन क्या फीचर्स हैं, which you will find in any life form. The cell membrane, it is not just a protective covering, there are several other functions also. We'll take up cell membrane. Like I said, I have to even cover plasma membrane separately. The third common feature here is the presence of a gelatinous matrix called as cytoplasm. It is again a site of various functions. And finally, both prokaryotes and eukaryotes have ribosomes, which are structures responsible for protein synthesis. Abhi tak, I hope I'm not talking Latin French. Abhi tak it's all things that you all know, right? Ayush Verma? Ayush, unmute yourself.
Yes, ma'am.
भव्या? हाँ आयुष, अभी तक ये सारी टर्मिनोलॉजी कोई भी ऐसी थी जो कि नहीं पता थी?
नहीं मैम, थोड़ी सी पता थी, मतलब पता ही है।
थोड़ी पता थी, ठीक है। तो अभी आने वाले हैं, अभी और भी बहुत टर्म्स आने वाले हैं। आई कैन नॉट हेल्प, जब तक शुरू के 1-2 क्लास में यू विल हैव टू अक्वेंट योरसेल्फ विद ऑल दीस टर्मिनोलॉजीज।
ओके।
नाउ Bhavya Agarwal, can you tell me what is the central dogma of life? Or have you heard about the central dogma?
Central dogma, nucleus.
क्या nucleus? Central dogma of life. Anybody? Raise your hands if you know it. Dipanshu Verma. चलो ध्वनि tell me.
Ma'am, DNA goes to RNA goes to protein is the central dogma.
नहीं, goes to मतलब DNA goes to मतलब क्या होता है?
DNA is converted to RNA and RNA is converted to protein.
नहीं, it's not. विशांतन, converted नहीं होता है विशांतन.
Am I audible?
Yes, you are. Tell me.
Ma'am, basically, it's like the process in which like the flow of information takes place between various like forms, like from DNA to RNA and RNA to protein, like through transcription and translation.
ठीक है, देवप्रिया.
Yeah,
बता दिया उसने. Same thing, हाँ. So why I mentioned the central dogma is it is not just the structures which are common in prokaryotes and eukaryotes. There are certain processes which are also common in both these cell types, and central dogma happens to be one of it, which states that the information which is present in the DNA flows through the RNA and finally forms functional proteins. This is a unidirectional mode of transfer of information. And the two steps, like you said, Vishantan and Devpriya, they are called transcription, which is flow of information from DNA to RNA, and translation, which is the process of forming proteins from RNA to from RNA. This is a common feature for all cells. तो कौन मुझे कह रहा था कि ये कन्वर्ट होता है? एनीवे, कन्वर्ट नहीं होता, ये इन्फॉर्मेशन रीड होती है. डीएनए की जो इन्फॉर्मेशन है ये रीड होती है आरएनए में, और उसके बाद से फंक्शनल प्रोटीन्स बनते हैं. एनी सेल, all the activities that take place in a cell is because of enzymes and proteins, right? So basically this is how the makeup of the cell or the potential of the cell is decided. This remains a common feature for both prokaryotes and for eukaryotes. Now here is a summary figure depicting the difference between a prokaryote and eukaryote. During your schooling you would have typically studied a eukaryotic cell. I will be describing the prokaryotes first, and in subsequent lectures you will be helping me figure out what is a eukaryote. From this figure, what is the difference that you can make out between the prokaryote? Two very prominent differences can be noted here. What is it that you can point out here? Raise your hands, please. Vishantan?
Ma'am, can you please repeat the question?
I'm saying, just from this figure, can you point out some major differences between pro and eukaryotes? Vishantan, answer कर रहे हो या फिर हम बाकी लोग जो hands raise कर रहे हैं उनको बोलें?
Ma'am, basically, like we can see in the prokaryotic cell, the genetic material is just suspended in the cytoplasm. It doesn't have any proper structure around it, like enclosing it, like in case of eukaryotic cells.
ठीक है अभय, thank you. Soumya, anything else?
Yeah, as you mentioned, absence of nucleus and other
उसके
cell
अलावा बोलो,
organelles.
बाकी लोग hands फिर lower करो जो सिर्फ ये कहने वाले हैं. उदिता?
The cell wall, prokaryotic has a cell wall and eukaryotic does not have it.
Yeah. Kashish?
The eukaryotic cell has various cell organelles, whereas
Yes.
prokaryotic cell doesn't.
Yes. Good. Abhay, कुछ और कहना है?
Yeah. Eukaryotic does not have cell wall and prokaryotic have cell wall.
ठीक है, तो कुछ न्यूक्लियस या प्रो और यू जो मेजर डिफरेंस था कैरियन के कॉन्सेप्ट में, उसके अलावा देयर आर टू थिंग्स दैट यू नोटिस हियर. An elaborate cytoplasm in eukaryotes, which is impregnated by a number of different structures, which you see is missing in the prokaryote. The prokaryote you see is more simplistic. But if you notice, the prokaryotic cell has multiple layers beyond the cell membrane or plasma membrane, which is not present in the eukaryotes. We will see what these additional layers are. Now, coming straight to prokaryotes, the main points that differentiate, I've just jotted out some of these points, besides the absence of a true nucleus like we saw, is that it's relatively smaller in size compared to eukaryotes. Now in biology, you have to always keep in mind that there can always be exceptions. We will see at places wherever there's appropriate, we'll talk of exceptions as well. Next, as you saw in the figure, there is minimal what is called as compartmentalization. That is, formation of compartments which are dedicated for different functions. So there is lack of organelles in prokaryotes or lack of compartmentalization. They also do not have cytoskeletal elements as found in eukaryotes. ये क्या होता है? Cytoskeletal elements क्या करते होंगे? किससे होते होंगे eukaryotes में? आपकी बॉडी में skeleton क्या करता है? हाँ जसकीन?
Provide a structure to the cell.
Yes, it provides a framework.
Framework.
है ना? So eukaryotes have a very distinct cytoskeletal elements, and it was believed that prokaryotes lack cytoskeletal elements. However, when we talk in detail, you will see that there are certain analogies that you have in prokaryotic cell as well. Then there is absence of typical cell divisions which are found in eukaryotes, that is mitosis and meiosis. How many of you have seen onion root tip cells dividing or have seen stages of mitosis and meiosis in your school time? Raise of hands please. Akshat Jain और कोई? चलो ठीक है. तुम लोग डेफिनेटली बायो में तो पढ़ोगे ही अपने इंट्रोडक्शन टू बायोलॉजी में. वी विल सी कि क्या डिवीजन, कौन सा डिवीजन प्रोकैरियोट्स में होता है. एंड शायद मुझे ब्रीफली फिर तुम्हें बताना पड़े कि माइटोसिस मेयोसिस क्या होता है, बट जब वो टॉपिक लेंगे, जब हम यूकैरियोट्स पढ़ेंगे तब करेंगे. फाइनली देयर इज नो सेक्सुअल रिप्रोडक्शन इन प्रोकैरियोट्स. अब मुझे ये बताओ कि सेक्सुअल रिप्रोडक्शन के एब्सेंस या प्रेजेंस का क्या सिग्निफिकेंस है? व्हाट इज द significance of sexual reproduction? What actually happens? Raise your hands. हाँ Bhavya.
Two partners are needed for reproduction.
ठीक है, उससे होता क्या है? What is the resultant of a sexual reproduction? There are two partners, the gametes fuse, there's a zygote which is
Genetic material is combination
हाँ
of the
Bhavya?
both.
Sorry?
The resultant genetic material is combination of both parents.
Okay, Vishanta?
Ma'am, basically, as Bhavya said, the cells basically exchange genetic material, so there is differentiation that occurs in the progeny.
Correct.
So
the source of variation in eukaryotes happens to be sexual reproduction. Why is variation needed? That is important for evolution. You have to evolve, and so you need certain variation. When I say that prokaryotes do not undergo sexual reproduction, you have to understand that the division or their progeny will always be identical to the mother cell. So there will be no variation per se when they divide. Okay. So over the next two lectures, we'll see that some of these points really do not hold true. And there's a level of compartmentalization, and even some cytoskeletal elements are found in prokaryotic cells. So this slide does not entirely stand true as of now. is how a typical bacterial cell looks like, wherein you easily observe the genetic material that you see, which has been termed as nucleoid. The cytoplasm is impregnated with these bead-like structures called ribosomes. The cell membrane has additional coverings like the cell membranes that is beyond the cell membrane. You have coverings like the cell wall, the capsule, and there are certain appendages that you see. There are pili and flagella that you find. Now, here is again a comparative figure showing the position of a typical bacterial cell on a size scale compared to eukaryotic cells and biomolecules. I had shown a similar one in one of my earlier lectures. So to understand the details of cell structure of a prokaryote, you can understand that you need to have an electron microscope to study the further details of the structure. With respect to its size and shape, prokaryotes have a larger surface area to volume ratio compared to eukaryotes. Such a larger surface area to volume ratio ensures faster rate of movement of nutrients. And this correlates with prokaryotes having a higher metabolic rate, a higher growth rate, and thus a smaller generation time as compared to eukaryotes. If you remember, this is also one of the reasons we discussed for their success in one of our earlier classes. Now this figure shows you how, keeping the volume constant, you can see that the shape actually determines the surface area for a cell. So from a sphere to an oblong cylinder, how the surface area per volume changes. What do we mean when we say that they are smaller than eukaryotic cell? I am presenting to you the size range which has been observed for some of the prokaryotic cells. While the model system that is popularly used to study bacteria, that is Escherichia coli, has a typically cylindrical shape of 1 to 2 micron, there are organisms like spirochetes which are approximately 500 micron in length. In fact, we now know that as an exception, there are also prokaryotic cells which can be seen by naked eyes with the dimensions of approximately 3 or 4 millimeter. Now, we talked about what is the definition of microorganisms, and we said we cannot see them with naked eye, right? But these are the maximum sizes that have been reported. If you look at the other extreme, you will see that mycoplasmas are considered to be the smallest prokaryotic cell of approximately 0.25 micron. Now, there is a very controversial group of bacteria called as nanobacteria, which are believed to be, some believe them to be the smallest replicating bacteria with dimensions in nanoscale. But there is a lot of controversy attached to it, so I will leave it at this. I have added towards the end of the PPT, I've added a news article talking about what this controversy is. That was about the size. Now talking of the different shapes of prokaryotic cells. The most common ones are spheres, also called as coccus or cocci, that's the plural. And the next common shape is rods or cylinders called as bacillus or bacilli. Now sphere, as you know, has the minimum surface area to volume ratio. We saw it in the last slide as well. This confers various advantages, including for intake of nutrients. But it's detrimental in certain cases, like cells in a desiccating environment where they want to lose water through the surface. It is there that you have the spheres at a disadvantage. The rods of the bacilli have the advantage of having a streamlined body for movement, and of course they have a larger surface area to volume ratio. There are various speculations as to why they exist in different shapes and if there is a significance attached to it. In fact, Professor Preeti from our department works a lot on how cell shape is affected by different genetic makeup and different environmental conditions. Here I have to mention that the arrangement of these spheres or rods is also in a specific manner and has a terminology. As you see, when two spherical cells come together, they're called as diplococci, while a string of cocci is called a streptococcus. And on the other hand, a staphylococcus is when cells divide in all plane to give a cluster or a clump of cells. Then there are other shapes, like you have the comma shape called as vibrio. Even spiral cells are present. And there's some very specific shapes like the star shape or the crescent shape. Some bacteria, however, are pleomorphic, which means that they can switch between different forms in response to changing environment. And a perfect example of this is mycoplasma, which I mentioned as the smallest bacteria. Now, let's start with cytoplasm, which I mentioned is a common feature for prokaryotic and eukaryotic cell. It has a gel-like consistency and is majorly made of water. It is the site of various functions and is especially of significance in prokaryotes because unlike eukaryotes, they do not possess the specialized compartments for different functions. So it is this matrix where most of the action takes place, whether it is metabolic reactions or steps involved in the flow of genetic information. Cytoplasm is the genetic material of a prokaryotic cell, which happens to be the DNA, the double-stranded DNA molecule, packaged in the form of a chromosome. Now, the prokaryotic chromosome is circular, and there's only, Akshat, ask me. Okay. The DNA that forms the prokaryotic chromosome is circular, and there's only one chromosome found in cells. The zone in which it is concentrated, the chromosome is concentrated in the cytoplasm, is called as nucleoid, which unlike a nucleus does not have a membrane around it, hence the primitive nucleus you saw. Now, how is the DNA organized? If you stretch the DNA from a prokaryotic cell, and this number is actually referring to E. coli DNA, its contour length would be approximately 1.6 millimeter. This is much larger than a typical bacterial cell, so it's forced to adopt a supercoiled structure to pack into a micron scale cell. And this packaging or compaction, it's not random, otherwise they'll get entangled. Like the eukaryotes, the DNA of prokaryotes is associated with a number of special proteins involved in maintaining the chromosomal structure. These proteins, one of which is the HU protein, that is the most abundant, are positively charged and they interact with the negatively charged DNA in the nucleoid. Now the compaction is also dynamic. So if you see in the figure, there are much more loops found in the cell when it is undergoing exponential growth, rapidly when it's growing, while a rather relaxed DNA is present in a cell that is in the stationary phase. The size of the genome of prokaryote, it varies from 0.5 megabases in the smallest cells like mycoplasmas to more than 10 MB in some species of cyanobacteria. There are certain exceptions, like for example, Bacillus megaterium has a genome size of 30 MB. But in general, most genomes are less than 5 MB in size. This is a comparative of the chromosomes of prokaryotes and eukaryotes. Unlike a single circular chromosome in prokaryotes, eukaryotes have multiple linear chromosomes. I'm sure you've come across human chromosomes as seen here. I mean, this kind of a structure which is linear. Akshay? Akshay, unmute yourself. Mute karo, matlab, mute kiya hua. Another important point of difference is that because there is no compartmentalization and no nucleus per se, the steps of transcription and translation, जो भी central dogma में देखा था, these take place simultaneously in prokaryotes, while in eukaryotes there is both spatio and temporal separation of both these steps, transcription and translation. Further, prokaryotic cells also happen to have extra chromosomal genetic material in the form of plasmids. Plasmids के बारे में सुना है? किसी ने? Anybody wants to tell me about plasmids? ठीक है, ये क्या होते हैं? They are extra chromosomal genetic material, which means genetic material or information in addition to the chromosome. They are also mostly circular DNA molecules, but much smaller in size compared to the chromosome. While they are not required for the general well-being and normal metabolism of the cell, they confer some selective advantages to the cell. अभी देखते हैं क्या क्या selective advantage confer करते हैं. They can replicate independent of the chromosome. If you remember in one of the last classes, I mentioned that bacteria can be manipulated or directed to synthesize particular metabolites of interest or maybe even change their features. For such approaches, plasmids have been widely used as vectors to introduce, let's say, the gene of our interest in the bacterial host. Like I said, they offer a selective advantage to the cells. Now, I have to make it clear, it is not that under normal situation or in all bacterial cells, you will have plasmids. They offer a selective advantage to the cells which harbor a plasmid based on the property that they possess. Now, there are various types. For example, the R plasmid, it offers resistance to, Ayush, bolo?
Can you please lower the call box? Shift call box. This is hiding the content.
Yeah, wait, I'll come to call plasmids and then, okay. I have to see that there's a message. I mean, if I don't see a notification here, then I don't know that you've raised your hands. Anyway, so the resistance plasmid or the R plasmid, like I said, offers resistance to several antibiotics. Those bacteria which possess the R plasmid will be able to grow despite the presence of antibiotic or despite an antibiotic stress, while those which do not have the plasmid will die, will eventually die. This is what I mean when I say selective advantage. We'll be talking about conjugative plasmid, that is the F factor or F plasmid, when we cover bacterial genetics. There are certain metabolic plasmids also of interest. If you notice, they can be utilized for bioremediation, wherein bacteria eat up pollutants in the environment, and these have been actually harnessed for cleaning up the environment. Other than the plasmids in the cytoplasm, like you saw in the earlier figure, there are ribosomes distributed throughout. Now, compared to eukaryote, the prokaryotic ribosomes are smaller. That is 70S ribosome is present in prokaryotes, while 80S are found in eukaryotes. Now, S here stands for Swedberg unit. We'll talk about it in a minute. There are two subunits, 50S and 30S in a prokaryote and 60S and 40S in a eukaryote. Please keep in mind that these units are not additive. Besides proteins, ribosomes are made up of RNA molecules, the ribosomal RNAs that you might know. The function of the ribosome, as I mentioned, is in the protein synthesis. Now, what is the Swedberg unit or the sedimentation coefficient? Because you come across this term for both proteins as well as for RNA. It is technically a measure of time and is equal to 100 femtoseconds. It is the ratio of the speed of a substance in centrifugal field to its acceleration, or how fast, basically in a comparable unit, of course. It is talking about how fast a particle settles to the bottom under centrifugal force. And this not just depends on the mass of the particle, but also its shape. So it depends on size, shape, and density of the particle. We said that the bacterial cell or the prokaryotic cell is devoid of compartmentalization. Lack of organelles, okay? But it's been realized that the prokaryotic cytoplasm is not as simple as it was earlier believed to be. There are a range of what is called as inclusion bodies depending on the type of bacteria present in the cytoplasm, which have specific functions. For example, There are inclusion bodies which serve the function of storage, like sulfur globules. Then you have glycogen granules. If you remember, I mentioned biopolymers. So that's PHB, polyhydroxy butyric acid, has been used as biopolymers. These are synthesized inside these globules or granules. Then there are inclusion bodies which serve other functions. Other functions other than the storage. For example, there are carboxysomes which store enzymes responsible for carbon dioxide fixation. Then there are inclusion bodies like phycobilisomes or chlorosomes, which are present in photosynthetic microbes, performing the function of harvesting light, as they contain light harvesting pigments. There are also gas vesicles. There are magnetosomes. So basically gas vesicles help maintain buoyancy, while magnetosome, another interesting structure that is found in prokaryotes, they help the cell align as per the Earth's magnetic field. We have covered all structures that can be found in prokaryotic cells in the cytoplasm. So please note that not all the structures are found in every bacteria. Okay. Having covered the structures in the cytoplasm, we'll now talk about the cell envelope, which is all the layers present in a prokaryotic cell starting from the cell membrane outwards. The prime function, of course, of the cell envelope is protection, and it's a complex structure. So you will realize this more as we talk about different layers, unlike what you saw in the comparative figure when I showed you a eukaryotic cell. The cell envelope is also involved in communicating with the environment. It maintains cell shape and rigidity, as well as it is involved in metabolism. A number of steps you will see activities occurring on the plasma membrane and beyond. In this figure, you can see that starting from inside out, the plasma membrane is followed by the cell wall, and then there is a capsule. Let's start with the cell membrane. How many of you are aware of the Singer and Nicholson model of or the fluid mosaic model given by Singer and Nicholson? कोई नहीं या सारे सो गए? Okay, no hands raised, huh? I will only briefly talk about it here because I have to anyway discuss the plasma membrane when we talk of archaea. The cell membrane is actually a unit membrane, as it has the basic structure in prokaryotes and eukaryotes. And even the compartmentalization that is present in eukaryotes, the membranes there also have a universal structure. It is made up of lipids and proteins. And I don't know if you've heard of it or not, but the structure, the most accepted model of the structure was given by Singer and Nicholson, called as the fluid mosaic model. This you will also cover in biochemistry, from what I understand, once you do lipids. A major difference between the cell membranes in prokaryotes and eukaryotes is the presence of sterols in the former, that is the eukaryotes. Now, what are sterols? Sterols are hydrophobic lipid molecules with ringed structure. Like cholesterol is a major sterol in vertebrates. And they have a role to play in maintaining membranes permeability and fluidity. Similar to eukaryotic sterols, some bacteria have hopanoids serving the same function. So hopanoids are also planar polycyclic hydrocarbons with a structure similar to sterols in terms of the rings that it has. In some bacteria, you will notice that the plasma membrane draws inside into invaginations which are believed to increase the surface area. Okay. Now, compared to eukaryotes, the function of plasma membrane in prokaryotes is pivotal, because in the absence of any dedicated organelle, it is the plasma membrane which is the site for major activities. So this is how the fluid mosaic model looks like. तुमने सच में फ्लूइड मोज़ाइक मॉडल नहीं पढ़ा है? हाँ, कोई मुझे बताएगा?
नहीं मैम, पहली बार सुना है नाम।
मैम
पढ़ा।
पढ़ा
पहली बार सुना है तुमने? तो
नहीं
स्कूल
मैम,
टाइम में नहीं हुआ ये?
मैम पढ़ा है
हाँ।
मैम,
नो मैम,
वी
नहीं
हैव
पढ़ा।
रेड इट।
पढ़ा है? आयुष ने पढ़ा है?
नो मैम, मैंने नहीं पढ़ा।
ठीक है आयुष, तो किसने कहा भी पढ़ा है?
मैंने भी नहीं
अभिनव
पढ़ा
ने?
था। नहीं, मैं बिल्कुल
शंकर,
नहीं।
हाँ बताओ विष्णु।
मैम
बिल्कुल नहीं पढ़ा है? अच्छा।
बेसिकली
हाँ
phospholipid bilayer पढ़ी थी, that there are like two layers of lipids which have
हाँ
hydrophilic and hydrophobic ends,
हाँ
and there are protein molecules embedded in between that, in between the two layers at certain junctures.
ओके तो वैसे तो प्लाज्मा मेम्ब्रेन मेरे कोर्स कंटेंट में नहीं है, मुझे बस उतना ही बताना है कि सेल स्ट्रक्चर में ये कैसे इंपॉर्टेंट है और कैसे आर्किया में, सो आर्किया बिकॉज दे आर एक्सट्रीमोफाइल्स, दे हैव अ वेरी डिस्टिंक्ट सेल मेम्ब्रेन व्हिच आल्सो हेल्प्स और कंट्रीब्यूट्स इन देयर सक्सेस इन एक्सट्रीम कंडीशंस, बट maybe I'll give you some brief when I talk about the cell membrane in the lecture on archaea. जो तुम्हें दिख रहा है अभी यहाँ पे, so basically it is a bilipid layer, हाँ? It is a bilipid layer and the lipid here is phospholipids. So the hydrophobic, so this is amphipathic molecule. These lipids are amphipathic molecules with both a hydrophobic and a hydrophilic region, and you will see that while the hydrophilic regions are on the outside, the hydrophobic region is hidden inside basically. And other than this lipids that you see, these globular hydrophobic parts that you can see, you have some proteins. So it's lipids and proteins which make up the plasma membrane, and the proteins are of two types. They are integral protein, the proteins which span the entire layer, if you see the bilipid layer, and there are also some peripheral proteins जो कि they are present on either the side of the plasma membrane. ठीक है? तो अभी हम और डिटेल नहीं बोलते हैं, जब हम आर्किया की बात करेंगे तभी, जब हम व्हेन आई वाज टेलिंग यू कि इट इज 40% फॉस्फोलिपिड एंड 60% प्रोटीन, दिस इज व्हाट इज मेंट, इट्स अ लिपिड layer. And when I said they do not contain sterols, actually I don't know यहाँ पे, हाँ, so for example ये देखो, this is a hopanoid that I said which is present in the prokaryotic cell, and in the eukaryotic cell the function of the hopanoid is performed by sterols. तो ये एक तरह से they act as a barrier breaking the fluidity which is created due to the lipids. लेकिन I will wait for I will take up archaea only once you've done the plasma membrane in detail in your biochemistry class, and then one of you has to tell me कि lipids हो गए हैं, then I'll take up the plasma membrane of bacteria and archaea and a comparative of it. Okay. I would not like to take up the next layers क्योंकि उसके बाद midway मुझे छोड़ना पड़ेगा. Anybody has a question? I now see that Ankit has messaged. मैंने कहा हम मैसेज नहीं देख पाएंगे साथ में तुम लोग के. हाँ अंकित, ऑर्गेन्स आर वेल डिफाइंड इन यूकेरियोट्स, फाइन. अभी तक हमने जितने स्ट्रक्चर्स किए हैं उसमें किसी को भी कोई क्वेश्चन है, कुछ डाउट है, प्लीज मुझसे पूछो. देखते हैं, टेल मी.
मैम वो आपने डिफाइन किया था, बैक्टीरिया होते हैं सर्टेन जो चेंज कर सकते हैं अपना शेप, अमीबा को कह सकते हैं वो?
अमीबा बैक्टीरिया नहीं है देवप्रिया, इट्स अ यूकेरियोट.
ओके.
है ना? अमीबा तुम लोगों ने शायद पढ़ा होगा, उसका न्यूक्लियस और ये सब देखा होगा जब तुमने पढ़ा होगा. पैरामीशियम किया होगा, यूग्लिना किया होगा, स्कूल टाइम में किया था देवप्रिया ये सब,
यस मैम.
है ना? तो ये सब में इसे टिपिकली स्कूल टाइम में आप यूकेरियोट्स पढ़ते हो जिसमें बहुत क्लियर न्यूक्लियस दिखता है आपको, और बाकी ऑर्गेनेल्स, माइटोकॉन्ड्रिया पढ़ा होगा तुमने या कोई भी और स्ट्रक्चर. तो अमीबा नहीं, बट अमीबा का डेफिनेटली स्यूडोपोडिया जो तुम कह रहे हो, इट हैज अ शेप व्हिच चेंजेस. हाँ विशांतन?
I have a doubt in this very slide ki why is it like that ki during exponential growth phase the genetic material constricts a lot and during stationary it's relaxed.
ठीक है, वेरी गुड. वैसे तो हम लोग को बैक्टीरियल ग्रोथ में और बैक्टीरियल जेनेटिक्स में ये देखना है कि ऐसा क्यों होता है. तुम ये जो सर्कल्स देख रहे हो ना, आरएनए पॉलीमरेज और ट्रांसक्रिप्शन फैक्ट्रीज, ये बहुत ही एक्टिव जोन है. यहाँ पे सेल्स डिवाइड कर रहे हैं, दे हैव टू डिवाइड. ओके, सो द actually there is a lot of negative supercoiling imposed where the division or replication is taking place. पूरे के पूरे genetic material को replicate करना है so that it then goes into the next cell, the daughter cell, right विशांतन?
Yes,
तो
ma'am.
ये तो ना ये सिर्फ एक पोर्शन एकदम स्ट्रेन्ड होता है जहाँ पे इस डबल स्टैंडर्ड स्ट्रक्चर को ओपन करना होता है, जहाँ पे रेप्लिकेशन हो रही होती है. ठीक है,
Right.
तो बाकी जगह नेगेटिव सुपर कॉइल्स आ जाते हैं. इमेजिन अ स्प्रिंग, हाँ, जिसमें कि तुम या दो स्प्रिंग्स आपस में एंटैंगल्ड है और तुम खोल रहे हो उनको, ठीक है, या वूल फॉर एग्जांपल, you try to separate out two strands of wool and it will lead to a lot of strain on both the flanking sides, right?
Yes, ma'am.
है ना, एग्जैक्टली वही चीज होती है. लेकिन हम लोग जब ये रेप्लिकेशन कवर करेंगे तो तुम देखोगे कि एक्चुअली हो क्या रहा है. ये सारे ना फोक्स हैं जहाँ पे बहुत स्ट्रॉन्ग एक्टिविटी चल रही है, बहुत ज्यादा एक साथ साइमल्टेनियसली. और अदरवाइज when it's in the stationary phase there is no replication happening, it's more relaxed, you don't see these negative supercoils that you see in the left side. ठीक है,
Okay. Thank you, ma'am.
anybody else? बाकी सब कुछ क्लियर है तो ना. आज क्या क्या स्ट्रक्चर्स देखे, कोई एक? आरिश, आज तुमने कुछ नहीं बोला, बताओ आज हमने कौन कौन से स्ट्रक्चर्स कवर किए? अच्छा, आज आरिश है ही नहीं. ओके, और कौन? कौशिक?
Yes, ma'am.
हाँ कौशिक, टेल मी आज क्या क्या कुछ नए स्ट्रक्चर्स कवर किए या ये सब में से कुछ जानते थे तुम?
मैम प्लाज्मा मेम्ब्रेन के बारे में
पता
नया
था?
नहीं
अच्छा, नया
पता.
था. अच्छा, नहीं पता था. ओके, राइबोसोम्स जानते थे?
यस मैम, उसके बारे में जानते
सेंट्रल डोग्मा जानते थे?
ये जो क्रोमोसोम ऑर्गेनाइजेशन ये भी नया था.
अच्छा, क्रोमोसोम ऑर्गेनाइजेशन तो हम लोग ने किया नहीं, लेकिन इनफैक्ट वो तुम लोग मॉलिक्यूलर बायोलॉजी एंड जेनेटिक्स में ज़्यादा डिटेल में करोगे, बट फिर भी डिफरेंस बिटवीन प्रोकैरियोटिक एंड यूकैरियोटिक क्रोमोसोम समझ में आया?
यस मैम.
ठीक है. कैन आई आल्सो हियर सम गर्ल्स स्पीकिंग? राधिका? किसका माइक इशू है? ओके, और कौन है? उदिता?
Yes, ma'am.
What else? What are the other structures? For example, did you know of inclusion bodies?
No, I didn't know it before.
So now when we say कि prokaryotes में compartmentalization नहीं होता है, will you support this statement?
Sorry, ma'am, pardon.
The difference that we started off with was कि prokaryotes में there is no compartmentalization, right? Or there are no organelles. Now having known about inclusion bodies and about other structures, the plasma membrane invaginating, do you think it is devoid of compartmentalization?
I don't think that the inclusion bodies should be considered as compartments, but I'm not
Why
really
not?
sure. Because the inclusion bodies that we read
Why
about
not, Udita?
were mostly the chemical bodies, right? Like some sulfur globules and some things like that.
Anybody? Why should inclusion bodies not be considered as compartmentalization in prokaryotic cell? Rudransh Chaudhary. कोई बताएगा? पारस? हाँ विशांतन?
Ma'am, can we say that they aren't present in like they're present in cell on a case-to-case basis? Like it depends on the environment, and it's possible that in a similar cell in a different environment they might not be present, but that won't be the case for eukaryotic organelles.
Yes, you can say so. Bhavya, anything to add?
They don't have plasma membrane.
Yes. Compartmentalization या true organelles आप कहते हो जब they are enclosed in a membrane. Usually all when you see the mitochondria, when you see the chloroplast, or when you see any other structure, endoplasmic reticulum, Golgi complex, they are all enveloped by the unit membrane. So when you say compartmentalization or the presence of organelles, these organelles have, majorly all of them, you will see there are some exceptions though, have a membrane, a plasma membrane. So इसलिए inclusion bodies and like Vishantan said, these are only, it's only on a case-to-case basis, on an environment-to-environment basis that these bodies might be present. So you don't call them as organelles at all. You just say structures present or inclusion bodies present. This is one of the prime reasons why you still say that prokaryotes are devoid of compartmentalization. However, there are dedicated structures for particular functions. Okay, any further questions? If not, then I will I'll upload the lecture. ठीक है. Thank you, class.
Thank you, ma'am.
Thank you, ma'am.